this science includes many branches, such as medical microbiology, soil microbiology, water and food, industrial microbiology, and others. But most important branch is a medical microbiology. It's the branch of microbiology that deals with the microorganisms that causes disease for human and other organisms, and can be divided into several branches, including bacteriology, virology, parasitology. When we talk about parasitology, this means protozoa, not helminth. Mycology and immunology. Now, microorganisms belong to the family Protista, biological kingdom, including some eukaryotes and prokaryotes, virus, viroid, and prion. These are classified according to their structure, chemical composition, biosynthetic, and genetic organization. Now, how can we differentiate between prokaryotic and eukaryotic cells? One of the most important features that help and distinguish between two types of cells is the presence or absence of a nuclear membrane or true nucleus. Prokaryotic cells have no organelles, which means no mitochondria or Golgi apparatus, have a membrane, no have membrane enclosed nucleus, and no have histone. Histone is a protein that presents in the DNA that help in the supporting or give supporting to the eukaryotic DNA. Also, they contain complex phospholipids, sphingolipids, and sterol. This presents in rare cases. But most important feature is the absence of a nuclear membrane. So the DNA is spread. So the DNA of bacteria is spread in a specific region of the cytoplasm called a nucleoid. Have 16S, have 70S ribosomes, have a cell wall composed from peptidoglycan containing uronic acid. Peptidoglycan presents only in bacteria, but not in other organisms. Also, haploid have a single circular chromosome, have short-lived unprocessed mRNA. Processed mRNA presence in eukaryotes because the mRNA of eukaryote should be passed through the nuclear membrane and should be give supporting and the presence of intron, which should be splicing during a transcription. Have coupled transcription and translation. Because of the DNA of bacteria is a presence in the cytoplasm and not the presence of a nuclear membrane, so mRNA is entered directly from transcription and captured by ribosome, then translation is done and then protein synthesis. While eukaryotic cells contain organelles such as mitochondria, Golgi apparatus, and the nucleus bounded by nuclear membrane. This means true nucleus. These contain complex phospholipids, sphingolipids, histone in the DNA, and sterol. They lack cell wall. Plant cells and fungi have a cell wall, but not contain peptidoglycan. Peptidoglycan is specific for bacteria and have multiple diploid chromosomes, such as human have 46 chromosome, have relatively long-lived mRNA due to this mRNA is processed by gapping of 5' end and polyadenylation of 3' end and splicing of intron, because intron is non-coding region, exon is a coding region. Have 80S ribosomes and uncoupled transcription and translation. when mRNA of eukaryotic cells is transcribed inside the nucleus, this should be processed and then passed through the nuclear membrane to reach to the cytoplasm and captured by ribosomes. Now, there are several types of bacteria, including typical bacteria. Typical bacteria have a normal peptidoglycan, may be normal flora or pathogenic for the human, do not have sexual growth cycle, however, some can produce asexual spore, can be reproduced by binary vision. The second is mycoplasma. Mycoplasma rickettsia and chlamydia called unusual bacteria. Mycoplasma are the smallest and simplest types of bacteria that are self-replicating. Lack cell wall are only prokaryotes that contain sterol. Now, lack cell wall, this means no peptidoglycan. So any antibiotic that inhibit or kill bacteria by acting on the cell wall or peptidoglycan biosynthesis, not effective against mycoplasma, such as beta-lactam drugs. Beta-lactam drugs is a large group of antibiotics, including penicillins, cephalosporins, carbapenems, penicillins such as ampicillin, amoxicillin, cephalosporins such as cephaloxime, cefotaxime, cefotriaxone, carbapenem such as imipenem and meropenem. All these antibiotics is not active against mycoplasma because mycoplasma have no cell wall. This means no target for these drugs in bacteria. Also, this bacteria have a sterol in their cell wall. So cholesterol should be added to the selective media that help in the isolation of mycoplasma. Mycoplasma pneumoniae is a causative agent of atypical pneumonia. Third is rickettsia organism are obligate intracellular bacteria that are incapable of self-replicating and depend on the host cell to gaining their ATP. Chlamydia, bacteria-like obligate intracellular pathogen with a complex growth cycle involving intracellular and extracellular forms. This is called reticulate and elementary body. This is a specific and unique feature to the chlamydia. Chlamydia only bacteria that have a developmental cycle composed from two forms. Other bacteria reproduced by binary vision. Also depend on the host cell for ATP production. Now we study the Koch postulates that help in dividing the bacteria or microorganism that causes the disease. The first postulate is the organism must be found in a human with the infectious disease, but not found in healthy one. This is clear. The second is the organism must be isolated from human with the infectious disease and grown in a pure culture. This point or postulates have some confusion because of some microbes or bacteria cannot be isolated from patients and cannot be grown in a pure culture, such as Treponema pallidum that causes syphilis. This bacteria very fragile and has a long generation time, about 29 hours. so cannot be growing or isolation from patient. Third postulate, the organism isolated in a pure culture must be initiated disease when re-inoculated into susceptible animal. Also, this point or postulates have some confusion. This including, according to the second postulate, some organisms cannot be cultured or isolated. Also, some bacteria is specific for a human, can be caused disease only in a human, so cannot be injected or causes disease in susceptible animals such as mice, rat, or guinea pigs, like Bordetella pertussis that causes pertussis. This bacteria causes disease only in a human because of the receptor of this bacteria is present in the respiratory tract of human, but not in respiratory tract of mice, rats, or guinea pigs. The fourth postulate is the organism should be isolated from the experimentally infected animals. Now we study the structure of bacteria. Size of a bacteria is about 0.5 to 2 micrometer. In diameter, they haven't organelles, non-nuclear envelope, have a single chromosome and sternate cell wall. Now, shape of bacteria. Bacteria may be spherical, called coccus, or rod-shaped, called bacillus, or oval, called Coccobacilli. Vibrio is curved. Spirillum is have a spiral shape. Also, the arrangement of bacteria is very important in diagnosis and in classification of bacteria, such as cocci may be arranged in chain and called streptococci, such as Streptococcus pyogenes is a principal cause of tonsillitis, or arranged in clusters, grape-like clusters such as Staphylococcus, including Staph aureus, is a pathogen that causes food poisoning, wound infection, and other types of infection. Bacillus may be single, such as enteric bacteria, such as E. coli that presents a normal flora and most common cause of UTI, especially in women, or presents as a chain like Lactobacillus, which presents as a normal flora in the vagina. Coccobacilli, including enteric bacteria and others. Now, bacterial structures including flagella, villi, capsules, plasma membrane, cytoplasm, cell wall, lipopolysaccharide that presents only in gram-negative bacteria, picoic acid, inclusions, and spores. Flagella help in motility, movement, and swarming occur with some bacteria spread to entire petri dish like Proteus species. This bacteria, when cultured in a specific part of blood agar or nutrient agar, spread to entire petri dish. Arrangement basis for classification. This flagella may be arranged as a monotrachus, presence of one flagella in one end. Lophotrachus is a tuft at or group of flagella at one end. Amphitrachus, presence in both ends, and peritrachus, presence around the bacteria. As shown this pictures illustrate the location of flagella in the bacteria. Villi, short protein appendage, similar, smaller than flagella, adhere to bacteria to surfaces. Antibodies will block the adhesion. This villi may be considered help in adhesion of bacteria to the surfaces and help in the colonization of bacteria inside the human body. Villi considered in some cases as a virulence factor for bacteria, especially Neisseria gonorrhea that causes gonorrhea. This can be classified into F. pilus used in conjugation to exchange of genetic information. Capsule or slime layers. The glycocalyx is a polysaccharide material or polymers produced by bacteria to the external surface and surround the bacteria, help in the protection of bacteria from the activity of antibody and immune cells. Also help in adherence of bacteria to the surfaces and prevent phagocytosis. Complement cannot penetrate this sugar. Cytoplasm of bacteria composed from 80% of water, 20% of salts and proteins. Help in the provided osmotic shock. DNA presence inside the cytoplasm is a single circular plasmid, also extra chromosomal DNA, circular, associated with antibiotic resistant and presence inside the cytoplasm. Also, there is no organelle such as mitochondria and Golgi apparatus in bacteria. Now, bacterial genetics. The genetic materials of bacteria is not surrounded by a nuclear membrane, nor does it contain a mitotic apparatus. It is generally called the nucleoid or nuclear body. Consists of polyamine and magnesium ions bound to negatively charged circular supercoiled double-stranded DNA. Small amount of RNA polymerase, and other proteins are present. Bacterial ribosome have a sedimentation coefficient of 70S and are composed of 30S and 50S subunits. This subunit composed from or containing 5S, 16S, and 23S rRNA respectively. Ribosomes are the sites of action of many antibiotics that inhibit protein biosynthesis, such as erythromycin, azithromycin, tetracycline, and others. Now, cell membrane is a bilayer phospholipid. Water can penetrate this structure. Flexible, not strong, ruptures easily. Osmotic pressure created by the cytoplasm is affected of the cell membrane. Cell membrane in bacteria have enzymes that help in metabolism and energy production of bacteria. Cell wall. This is very important structure and characteristic for bacteria. Cell wall, or according to the thickness of cell wall, bacteria can be divided into gram-positive and gram-negative by using gram-string. Cell wall composed from peptidoglycan polymer is amino acid plus sugar. This unique to the bacteria. Sugar containing an acetyl glucosamine and an acetyluronic acid, and bacteria have D form of amino acid used not L form, hard to break down D form. So many lysozymes cannot be affected due to the presence of deformed amino acid. As shown in this figure, this is a basic structural unit of peptidoglycan composed from 2 sugar, an acetyl glucosamine and an acetylmuronic acid, bound by glycosidic bond and peptide bond connected the amino acid glycine with lysine, alanine, glutamine, and alanine molecules. This structure of cell envelope of bacteria composed from cytoplasmic membrane, periplasmic space. Periplasmic space presence only in gram-negative bacteria and containing peptidoglycan. Peptidoglycan in gram-negative bacteria is composed from about 5 to 10 layers, while in gram-positive bacteria composed from 20 to 40 layers. So the cell wall of gram-positive bacteria is more rigid and strong than gram-negative bacteria. But gram-negative bacteria has an outer membrane. This outer membrane composed from phospholipid and lipopolysaccharide. This lipopolysaccharide containing core polysaccharide and O polysaccharide, or called O side chain, also presence of lipid A. This is responsible for toxicity. Lipopolysaccharide of gram-negative bacteria is also called endotoxin. Endotoxin presence in gram-negative, but not presence in gram-positive bacteria. So, lipopolysaccharide represents a virulence factor for bacteria and main virulence factor for some of this bacteria like Salmonella typhi. Salmonella typhi has a long chain of oligopolysaccharide or O polysaccharide that help initiation of strong immune response in patients with typhoid fever and lead to production of interleukin-1, which is considered endogenous pyrogen. Now, cell wall determine the shape of bacteria, strength to prevent osmotic rupture. 20 to 40% of bacteria and unique to the bacteria. Some antibiotic effect directly against cell wall, such as penicillins. Thick acid presence in the gram-positive bacteria only. Are water-soluble polymer containing ribitol and glycerol residue linked by phosphodiester bond, are found in gram-positive cell wall or membrane, are chemically bounded to the peptidoglycan or membrane glycolipids, particularly in mesosome. This contains important bacterial surface antigen determinant and lipotic acid help anchor the wall to the membrane. May account about 50% of the dry weight of gram-positive cell wall. This also considered a virulence factor because it helps in the attachment of bacteria to the body surfaces. A lipopolysaccharide, as we talked previously, function atoxic, kill mice, pigs, and human, and is a leading cause of septicemia and death in enteric during the course of enteric fever. Considered as a pyrogen, causes fever, heat resistant, hard to remove. Now, in the spore, some bacteria, such as Bacillus and Clostridium. Bacillus, including Bacillus anthracis, that causes anthrax, and Bacillus cereus, that causes food poisoning and other infection. Clostridium have many pathogenic species for humans, such as Clostridium botulinum, that causes botulism. Clostridium perfringens, a leading cause of gas gangrene. Clostridium tetanus, that cause tetanus. Endospore help the bacteria to resist heat, radiation, cold, and boiling for more than 1 hour. So the bacteria remain available after boiling for 1 hour. This spore can be destructed by using autoclave. This or spore, when bacteria form spore, become metabolically inactive and highly resistant to desiccation and heat. This spore possesses a core that contains many cell components, a spore wall, a cortex, coat, and an exosporium. When germinate under favorable nutritional condition, after an activation process that involves damage to the spore coat. Also, this structure helps in differentiation between some species of bacteria depending on the size and shape of spore. Spore may be present in the center of bacteria, or subterminal, or terminal. This, now, according to our lecture, there are most important aspects should be take your attention. The first is the differentiation or differences between prokaryotes and eukaryotic cells. You should be understanding postulates, and you should be understand the structures of bacteria and the presence of some structure in gram-positive, but not a presence in gram-negative, and functions of these structures. This is very important notes in our lectures. Also, you should be understanding the relationship between the presence of specific structures and the activity of or action of antibiotic. Antibiotics is a chemical compound that used to eliminate the bacterial infections. Now, my student, there are any questions?